Hello, everyone. It's Steve with Aptera Owners Club. There was a new podcast at Cause Artist that Steve Fambro was on. Most of it is him going over the history of Aptera and the aerodynamic design and things we've all heard before. There were a couple of interesting points. This is going to be so much of a longer video, and towards the end, it's like stuff that's not really Aptera related, but I found it very interesting, so I'll talk it over with you guys. This first part is Steve Fambro talking about why the solar cells were rotated 45 degrees, which is, they have the patent on that to help it bend more. But you can tell that he actually just liked he and obviously others just liked the aesthetics of it.
And there's something fortuitous that happened in doing that. So we had these cells, and we were trying to figure out how to bend them in two axes. We realized two things. If we rotate it, 45 degrees, then you can bend it sort of bend the diamond portion as opposed like bending a diamond as opposed to bending a square. I don't know if that makes sense, but if you just rotate it slightly, because
Right.
Yeah, so I had a video about this, and a lot of people think that it's there were a couple commenters that thought it was all kind of frou-frou, but it is like you can bend the corners of it more than you can bend the sides of it. That's for sure.
this is counterintuitive because most people, you look at university solar projects or things like that, they pack all the rectangular cells together. And we had also wanted to avoid that aesthetic. I wanted to like subconsciously trigger something in people's minds that was elevated from that. And so the idea of rotating it 45 degrees also looks like a diamond, so it just connotes something different than having if it were just rectangular cells. It's hard to maybe imagine in your head, but if you look at the vehicle now, just imagine instead of them looking like diamonds, if you were to rotate it and they look like squares, it'd be kind of awkward looking. Once we realized that we could pack that many on the vehicle and have it become an integral design element, then we said, 30, 40 miles a day is really meaningful. That's more than most people drive. And it would.
Okay, so yeah, there was two things. The bending thing, which some people are saying is not really a thing, but me playing with the a piece of paper, it does help a little bit, at least. And then two, they just like the aesthetics of it. They thought that the diamond shape pattern just looked better. So that's kind of interesting. Okay, here's the most important part of the podcast: is he's talking about funding, and that I think that's some new information in this podcast that I hadn't heard before that I think is the most important part.
And so we're starting to cut some tooling now. We're not able to go as fast as we want because we are still in our Series B fundraising. That's the one you don't see. That's not the crowdfunding. That's the one. That's us going out to the capital markets, dealing with institutional investors, and
Gotcha.
those are what require Chris and I to travel. He's in Europe right now. We both just got back from Switzerland. I have to go to Dubai this week. I just found out this morning, and so we are. I would say 4 or 5 big deals in between the Middle East, India, and Europe that we're working that should bring in the production money into the company. But while we're, the 150, $200 million, not only to fully fund the factory, but to increase production to 2 shifts and do all kinds of other things that we want to do with service. So that plan is in the process of being funded, and we are still using our crowdfunding to do whatever tooling progress we can to inch closer and closer to production. So we're not just keeping the lights on, so to speak. Everybody here has their hands in doing something every day that moves us closer and closer to production. But they really become unlocked once we close the Series B fundraising round, which I'm very hopeful we have some very positive momentum with that. It's just stuff I can't talk about publicly.
Sure.
Okay, so the big news is they are working 4 or 5 big deals in between Europe, the Middle East, and India. I briefly talked sort of off the record with Chris Anthony several weeks ago, and he was telling me how capital markets in North America were very tight, but that the markets in India, the Middle East, and Europe were loosening up. And so that's where they're headed. And looks like they have, according to Steve Fambro, 4 or 5 good leads that they're currently working on. They're in talks with them. And then another interesting thing he said is he mentioned 150 to 200 million dollars. That is much more than the $50 million that we were quoted earlier. So I don't know if that means that like $50 million is the floor that they need, and that's what they would get to do the tooling and stuff, but really to like hire up the staffing because HR is always the most expensive part of anything. So to fully maybe to fully staff up the factory, they're needing more like 150 to 200 million. So 50 million gets them the tooling and the supplies, and probably they can start rolling out like slowly. But once they get 150, 200 million, then they can just go to full production and get like the full complement of the workers and stuff that they need. And just to be clear, this 50 million that's always been quoted is in addition to the 21 million that the California grant gives them, the matching grant that they give them. And so their needs are a combination of that, the matching grant, and also some creative deals with like Red Viking and some of the other big suppliers to lease their stuff instead of buying it outright, which gives initially smaller capital costs. Okay, so the last part of so I mean anyway, that's very exciting news that they have 4 or 5 deals. Steve sounded very optimistic that they were going to close these deals, and he just said, they can't really disclose, who those people are, the specifics of it publicly at this time, which makes sense. All right. So the last part is Steve talked a little bit about his background, and it's something kind of personally interesting to me because it overlaps with my professional work.
Late bloomer, so I didn't go to university until I was almost 30 years old. I went on the GI Bill and was very, mission focused to get through school. And once I got hired before I graduated. I mean, I still graduated, but I had a job offer that I accepted my senior year, and was at a biotech company here in San Diego called Illumina. And so my girlfriend and I at the time, she was also a double E at electrical engineering. That's how we met. We decided, and she's from San Diego, so we're like, oh, it makes sense. Let's head to San Diego after graduation. This job was it was like a dream job. I was one of, I think, one or two electrical engineers in the whole company, and I was junior. I'd worked as a technician for a long time, for maybe 10 years or so. So I knew how to repair equipment. I didn't know how to design. I was an engineer. So I knew my way around a lab and how to fix things. What's like the PhD scientists would be like looking at an instrument, questioning the results or what's happening. I would walk around and see maybe it's just turned off, or like check the
Right. Yeah.
fuse, something like that. So this job was a dream job, and it kept me just fully engaged, every bit of my brain fully engaged in solving problems on how to make DNA. That's what these machines did that we designed. We were designing how to image the DNA, how to just all the lab automation and stuff. Just it was fantastic. And then we got to a point where we engineered ourselves out of a job, so to speak. The last machine that we built it was like thousands of times more efficient than the previous one. So it cost us virtually nothing to make DNA, which is a necessary part for when you're not doing genotyping, when you're doing SNP analysis or single nucleotide polymorphism. It's when they're looking for a targeted area of your genome. You need to produce the opposite DNA of what you think you're looking for, or the conjugate pair. So that's why we had to produce DNA for this whole process to work. We were able to then produce it thousands of times cheaper than anybody else in the market. So we crushed everybody in the marketplace. And the machine went on to run another 10 or 12 years after I left the company. The thing is, once that happened, then we just got kind of the engineering team broken up and parceled out to the company, and I got bored. And if there's one.
Okay, so he worked for Illumina, and Illumina is a company that is pretty well known in the biotech sciences. Illumina is a company that is one of the companies that does something we call next generation sequencing. And next generation sequencing um, is this method by which you can sequence large amounts of DNA and figure out what the sequence is or what's written in the DNA uh, through massively parallel processes to make it extremely cheap. So the original method of sequencing DNA is something called the Sanger method, and the Sanger method is named after Frederick Sanger, who won the Nobel Prize in chemistry twice. He won it for the first time for figuring out the protein sequence of insulin, and then he refined his DNA sequencing technique And so he got two Nobel Prizes. He's one of only five people in the history of the world that's gotten two Nobel Prizes, and only three people that have won multiple Nobel Prizes in the same category. Um, so that this is like every biochemist and chemist and biologist knows about Frederick Sanger and the Sanger sequencing technique, which works by a method where they extend the DNA sequence, and they have these special nucleotides that you can't extend anymore. So they just stop at each. They make different lengths, and then you run this on a gel, and the gel separates it by size. And if it has this color, then it's we know it's an A. If it's this color, it's a G, and things like that. But you have to like remake the sequence to read each position on the DNA, which is extremely expensive and time consuming. And that's why when they first did the Human Genome Project several decades ago, the human genome is about 3 billion base pairs, and I think they spent about $3 billion to do it. To sequence the entire human genome, it costs us $3 billion. Now, with this next generation sequencing, you can sequence an entire genome for about $1,000. So the cost of genome sequencing, because of companies like Illumina and what they did, went from $3 billion to $1,000. So that's pretty impressive. And this is a cool video, but that Illumina puts out that talks about
through reduced cycle
how they
amplification,
do it. And so
additional
we can watch it
motifs are
together if you're
introduced,
interested. If
such
this is
as the
not
sequencing binding site,
this
indices.
is not Aptera related, but it's very interesting to me. And so if anyone wants to know how next generation sequencing works, you can follow along with me and finish off the rest of this video.
And regions complementary to the flow cell.
So they extract the DNA and they chop it up, and they align these little indexing leads to either end of it. And so this is all done automatically. You just feed it into a machine, and this robot does the pipetting. It's a lot of manipulation of very small fluid volumes at very tight tolerances. And this is probably the kind of stuff that Steve Fambro was working on, like working on the robotics and the imaging of it.
Oligos. Clustering is a process where each fragment molecule is isothermally amplified. The flow cell is a glass slide with lanes. Each lane is a channel coated with a lawn, composed of two types of oligos. Hybridization is enabled by the...
So oligos are just very short sequences of DNA. So they're usually like 10, 12. And so these things will pair up with the little things that they added on to the sample prep, the sides of the sample.
First of the two types of oligos on the surface. This oligo is complementary to the adapter region on one of the fragment strands. A polymerase creates a complement of the hybridized fragment.
So in order to do this, they have to have the robot just pipette the sample on and then wait a certain time and then wash it off, the stuff that doesn't stick, and then they got to add the enzyme and the reagents to make it amplify.
The double-stranded molecule is denatured, and the original template is washed away. The strands are clonally amplified through bridge amplification. In this process...
So so this is really another step that the machine does, is they heat it up. When you heat up DNA, it separates from its two strands, like you basically unzip the ladder, and then you can wash away the things by adding more buffer.
The strand folds over, and the adapter region hybridizes to the second type of oligo on the flow cell. Polymerases generate the complementary strand, forming a double-stranded bridge. This bridge is denatured, resulting in two single-stranded copies of the molecule that are tethered to the flow cell. The process is then repeated over and over and occurs simultaneously for millions of clusters, resulting in clonal amplification of all the fragments.
So now you have a lawn of multiple copies of many different sequences in parallel throughout this whole thing.
Bridge amplification, the reverse strands are cleaved and washed off, leaving only the forward strands. The 3' ends are blocked to prevent unwanted priming. Sequencing begins.
Okay, so this is the cool part. So instead of like doing the Sanger sequencing where they extend the whole thing and each strand just gives you one read, they get a continuous read as it builds. So you'll see how
But
that
the
works.
extension of the first sequencing primer to produce the first read. With each cycle, fluorescently tagged nucleotides.
So for both of you guys that those of you guys that forget biology, the A's and the T's they stick together. The G's and the C's they stick together. So across from a T you can only have an A, and across from an A you can only have a T. Across from a C you can only have a G, and across from a G you can only have a C. And so they have different molecules of T's that are all tagged to a specific color of fluorescent probe.
Compete for addition to the growing chain. Only one is incorporated based on the sequence of the template. After the addition of each nucleotide, the clusters are excited by a light source, and a characteristic fluorescent signal is emitted. This proprietary process is called sequencing by synthesis.
So every time a new letter gets added, it sends a different color light to an imaging sensor. So every time it adds one, it gets a light, and you can say that, oh, that was a T, that was a T, that was an A, G, C, G. And so you're reading, like with Sanger sequencing, you would have had to make, like 8 strands to read this, and then 9 strands to make the 9th one. But you're doing this off of all one strand.
The number of cycles determines the length of the read. The emission wavelength, along with the signal intensity, determines the base call. For a given cluster, all identical strands are read simultaneously. Hundreds of millions of clusters are sequenced in a massively parallel process. This image.
So you can see each one of these dots represents one of those little strands, and as the color changes, the imaging sensor is like reading off what letter is being incorporated. And this is a small section, and you can see this is like thousands of strands. So it's reading millions of strands simultaneously.
Represents a small fraction of the flow cell. After the completion of the first read, the read.
Okay, so then they repeat the read multiple times, and then what happens is then you do just like data analysis.
The read 2 product is then washed away. This entire process generates millions of reads,
that are
representing all the fragments. Sequences from pooled sample libraries are separated based on the unique indices introduced during.
So you'll see that these things overlap, and the computers align the overlapping segments to make a complete single read.
For each sample, reads with similar stretches of base calls are locally clustered. Forward and reverse reads are paired, creating contiguous sequences. These contiguous sequences are aligned back to the reference genome for variant.
Yeah, so see, you have all these overlapping reads, and then you build them together, and it makes one continuous read of the entire thing. Anyways, the whole thing is completely ingenious and has completely revolutionized both biochemistry, molecular biology, and genomic medicine. And it's not possible without this highly automated sequencing technology that has become really, really cheap. Like if it cost $3 billion to sequence some person's tumor, you would never do that. But if it costs $1,000 to sequence someone's tumor, well, then that becomes something that's doable. All right, so very interesting that Steve Fambro worked for Illumina, which is a very well-known company in the biotech sector. And the most exciting thing is that they are working on 4 or 5 big deals, and sounds like they're hoping to get 150 to $200 million out of these 4 or 5 big deals. So that's very heartening to hear that. And the confidence that he had when he said it, he said they feel real good about it, just can't give details. And so it makes us feel good that they're well on their way to production. Okay, well, thanks for watching, guys. Thanks as always to our supporting members. Have a great day, everyone.